first of all, for your kind introduction. And let me thank, of course, all the organizer, Vitali, for allowing me to give a presentation today. So what I would like to discuss with you is what happened basically in the field of nanomedicine in the last 20 years or so. I mean, when I started my PhD up to now. And I think this long journey has been characterized by great success. If you can think of, for example, the very successful RNA vaccination that most of us use for COVID vaccines, as well as it has been characterized by disappointing failures. I mean, if we look back and, I don't know, maybe a lot of clinical trials that failed because this nanoparticle couldn't reach effectively the target of tumors that were designed for. And the challenges nowadays are mostly, are still the same. As you can see from these cartoons, it's a question of crossing several different biological barriers before reaching the cells, and in drug delivery, of course, delivering successful a drug to the cell we want. First of all, as soon as, for example, polymeric nanoparticles or liposome, whatever you like, are injected into the bloodstream, they need to avoid the immune recognition, they need to avoid opsonization, accumulation in the organs of the mononuclear phagocyte system. So clearly we have to think about the proper biodistribution. So we have to avoid the accumulation in the highly vascularized organs, typically, I would say liver and spleen mostly. But even if we can skip and we can prolong this circulation, then we need to find a way to extravasate, to diffuse into the extracellular matrix in order to reach the targeted cells. And in terms of extravasation and diffusion, of course, we have to design the particle size accordingly. But this is not the end of the journey. Of course, even when nanoparticles, the nanocarrier, can interact with the cell, well, this interaction has to be properly designed. Because in some cases, we need to induce endocytosis. For example, the nanoparticles needs to be uptaken, and then in most cases, it needs also to skip the endosome, delivering the payload in the cytoplasm. In other situations, we need to trigger specific cell activation by interacting with the membrane proteins of the cell. And the other additional barrier that sometimes we need to cross is the barrier that we can encounter in the nucleus in order to deliver, for example, DNA or other transcription factors. So depending on the payload, depending on the targeted cells we are looking for, clearly the design of the nanomaterial will change accordingly. So trying to overcome all this barrier, I start discussing about this project. In this case, we wanted to design a proper polymeric material that could enhance the circulation of therapeutic proteins. So therapeutic proteins have been widely used in pharmaceutics. Nowadays, there are several different therapeutic proteins which are commercially available. They can offer different treatments depending on the activity of the protein itself, but they also have some limitations because, for example, they can be often recognized by the immune system, as I said before. They could be hydrolyzed often quickly by the presence of enzymes, which are in a bloodstream. And if they are too small, they can also be excreted by kidney pretty soon. So in order to prevent all these drawback, at the same time enhancing as much as we can the circulation of these therapeutic proteins, we can definitely pegylate them. So the standard approach to achieve this characteristic is to attach a PEG molecule to the protein. As we know, a PEG is a typical protein repellent polymer, hydrophilic, biocompatible, and therefore it can definitely enhance the circulation of these compounds. On the other hand, we also know that polyethylene glycol can be recognized by some antibodies that we have, and therefore the circulation is not as much prolonged as we were expecting some years ago. At the same time, the typical grafting 2 approach, which is like grafting different PEG chains to these proteins, well, it may hinder the final activity of the protein itself, because sometimes this PEG chain can be grafted close to the active site of the protein. So in this, with this approach instead, we wanted to use a so-called grafting from approach. So which is basically like growing the polymer chain from a specific, starting from a specific group of the protein, which is supposed to be far away from the active site, first of all, and then using a typical control-even polymerization technique, such as atom transfer radical polymerization. In this case, that was particular argent ATRP, which can be carried out also in water, so without affecting the stability of this water-soluble protein, of course. We can definitely control the molecular weight of the chain, as well as using multiple initiators, multifunctional initiators, in order to get more than one chain. So we can design a much more complex architecture. At the same time, by using the same control-even polymerization, by simply changing the monomeric unit, we can vary also the chemical characteristic, chemical properties of the polymer itself. So with this study, basically what we did, we synthesized different ATRP initiators. As you can see from these schemes, we synthesized one single ATRP initiator or 2 functional ATRP initiator linked to an aldehyde group. Basically, the aldehyde can react very efficiently with the terminal NH2 of the protein. In this case, we use a lysozyme as let's say, model protein. And this conjugation can be achieved by a relatively simple reductive amination, simply by controlling the pH, it has to be slightly acidic, together with the reducing agents. And then by using the argent ATRP in buffer, as you can see, we can either polymerize PEG methacrylate, as well as this glycerol methacrylate, which is also known to be a typical protein repellent polymer with characteristics which are similar to PEG. So in other words, we can synthesize either a single chain polymer or a double 2-arm polymer, and we can vary again molecular weight and the composition. Of course, we proved that the reaction was definitely control-leaving. I mean, the kinetics followed a first order kinetics, and this is a semi-log plot. We proved that it was possible to achieve a chain extension, which means basically that we start a first polymerization, we purify the protein, we start a second polymerization, and the molecular weight keeps on growing as expected. But the interesting part was to study what was the effect of the grafted polymer to the activity of the protein. So, for example, we demonstrated that the relative activity of this lysozyme was above the 80–90% at its maximum when we use a glycerol methacrylate with a double chain rather than a single one. Perhaps because by having a multi-arm architecture, we can definitely have a more compact structure which is far away from the active site of the protein. On the other hand, if we look at the retain activity, so because we studied here was the stability of the protein after the polymer grafting, we saw on the other hand that the PEG methacrylate chains was much more effective in preserving the activity. And this perhaps because of the fact that the PEG chain, since it's like a brush polymer, PEG brush polymer, we may have a much more spherical hindrance to preserve stability. This has been proved both for serum, so incubation with the serum, which contains enzymes which can potentially cleave the protein, as well as the trypsin, which is another typical cleaving agent. And then we also proved the stability of the protein when heated at 90 degrees for 30, 60, and 120 minutes. And even in this case, we demonstrated the PEG methacrylate polymer was the most, the best one in terms of preserving the activity of the enzyme. We think that in this case, that was due to the temperature, which was close to the lower critical solubility temperature of the polypeg methacrylate. And in this case, a partial dehydration may further stabilize the tridimensional structure of the protein, even at high temperature. So to sum up, I just say that by, let's say, designing a proper polymer architecture and the monomeric unit, we can tune basically the final activity and stability of the protein. The question is now, okay, this is about proteins. What if we want to simply design a typical polymeric nanocarrier for delivering a low molecular weight drug, for example, to tumors or unhealthy tissue? In this case, I already talked about the problem of skipping the immune recognition, for example, avoiding opsonization. So we can definitely use a PEG layer for decorating the nanocarrier. But at the same time, as I mentioned at the beginning, at some point we need to extravasate and diffuse to the extracellular matrix if we want to reach the targeted cells. So from this point of view, we have to think in terms of size, and the nanoparticle size, again, from this point of view, I would say the smaller the better. There are experimental evidence, for instance, that nanoparticles which are in the range of 20 nanometers or so can diffuse, can also reach a lymphatic system, whereas nanoparticles as big as 100 nanometers, cannot. So definitely we would like to have a nice control over size. And from this side, micelles, polymer micelles could be the best option, because by a bit of simple polymer chemistry, so basically we just have to synthesize a block of polymer. So for example, I don't know, a block polyester and a polyethylene glycol, we can mix it with an hydrophobic drug, and we can easily make a suspension which can be injected potentially into the bloodstream. However, here comes the problem, because most of these polymer micelles present a critical micellar concentration, as you may know, which means that as soon as you inject this nanomaterial into the bloodstream, well, an adult has a blood volume of about 5 liters, so you get a gray dilution. And if we go down to the critical micellar concentration, we definitely dissolve all these micelles into unimers, and the drug will be released straight away without even reaching the target. So we can circumvent this problem by introducing a sort of a unimolecular structure or micellar structure, which means that we can synthesize amphiphilic copolymers, which as a single macromolecule, they already are a micelle, and therefore, even if they are diluted as much as we can, they're still a micelle and they can still encapsulate a drug. So we don't have theoretically this problem of a critical micellar concentration. And in this study, we evaluated what was the effect on the macromolecular architecture on the size of the nanoparticles, as well as the critical micellar concentration. So we can easily synthesize a polyepsilon caprolactam, so a polyester-based macromolecule. We can copolymerize still through ATRP with the PEG methacrylate, getting linear amphiphilic copolymer. We can generate a 4-arm version of it. We can generate the 4-arm version of a block copolymer with an inner core of hydrophobic polyester and outer shell of a polyethylene glycol brushes, or a sort of brush block copolymer, which has this architecture, as you can see here. And the synthesis is relatively straightforward. I mean, the only problem was to synthesize either polyester methacrylate precursors as macromonomers. The PEGMA is already commercially available, as well as caprolactam, polycaprolactam-based micro-initiators. So they were functionalized with this bromine groups here in order to get the further ATRP to synthesize the outer brush. So as I mentioned, by varying the architecture, we can control in a way the final size of the nanoparticles. The best results were obtained by using the 4-arm polymers. As you can see, the nanoparticles, this dynamic light scattering measurement, shows a typical size of about 10, no more than 20 nanometers. This is exactly what we were looking for. As I said previously, the 10, 20 nanometers nanoparticles are much more efficient in crossing biological barriers. When a linear chain, linear architecture was used instead, we can also see a sort of bigger self-assembled nanoparticles. And also the final size was also dependent on how large the hydrophilic p-PEGMA brush was obtained. So by increasing the degree of polymerization of PEGMA, we can have an effect in reducing the final size of the nanoparticles. In terms of critical micellar concentration, instead, we characterize it by a static light scattering. And as you can see, if we see the scattering intensity, there's a linear decrease by decreasing the concentration of the nanoparticle, which basically means that there's no critical micellar concentration as we expected. The only critical micellar concentration was found for the brush block copolymer configuration, where I think this transition at low concentration was mainly due to the dispersity of the polymer, because unfortunately by increasing the complexity of the architecture, clearly the final dispersity of the polymer, the molecular weight distribution can be affected. All right, so by controlling the size of the nanoparticles, we were also able to control the permeability of these nanoparticles, for example, in terms of renal excretion. So in this recent study, we of course, proved that graphene quantum dots, that they have a very small, you see, size distribution. I mean, the average size in here was only 3 nanometers, can definitely cross the glomerular filtration barrier of kidney as expected, because under physiological condition, this kidney barrier has a threshold of about 6 to 7 nanometers corresponding to the size of albumin, so that they can definitely pass through. But on the other hand, the typical polymer nanoparticles cannot, because again, the maximum threshold would be about 20, 30 nanometers, no more than that. On the other hand, by using our 4-arm structure as of our copolymers as presented previously, we were able to control the final size of the nanoparticles, getting them very small. So these bars correspond to a polymer with an hydrodynamic diameter of only 6 nanometers, this one 10 nanometers, and this other 25 nanometers. So as expected, I mean, of course, the smaller they are, the more they can cross the glomerular filtration barrier. These are milligrams of polymers that were collected finally in urine. So which means that the polymers was able to cross the glomerular filtration barrier, reaching the bladder. And of course, by increasing the size of the particles, we have a reduction of this permeability. But on the other hand, when the kidney in mice were damaged, we induced and damaged, mimicking chronic kidney disease, of course, this nanoparticles permeability increased, and this would be potentially useful for delivering drug, for example, to podocytes, which are dramatically affected by this sort of kidney diseases. Okay, then the other typical problem in nanomedicine is to characterize the biodistribution. I mean, you know that with polymer chemistry, we can do a lot of things regarding, you know, grafting, functionalization of polymers with fluorophores. We can use near-infrared fluorophores. They can be seen pretty well when we get images of small animals like mice. But of course, when we deal with humans, with patients, I mean, fluorescent emission is not sufficient to get a nice study of the biodistribution in vivo because of relatively low penetration depth of light. So we need to find alternative way to track our nanoparticles and try to identify where they go in our body. We can use, of course, a radioactive labeling, as in this picture, but we recently developed a method for attaching to our polymeric material fluorinated compounds. And by doing so, these final nanoparticles are traceable by using 19-fluorine MRI imaging, which can be a good option for using MRI in vivo in patients. So we use different possible approaches. The simplest one you can see, this is a collaboration that we have with the group in my department with Professor Baldelli, Bombelli, and Professor Metrangolo. We simply attach these fluorinated compounds to the end group of PLJ using a standard carbodiamide chemistry. Clearly, the final fluorine NMR show a good narrow peak, a sharp peak. And of course, these nanoparticles then, once formed, they can also encapsulate an hydrophobic drug, such as, for instance, dexamethasone, used in this study for treating cell cultures of podocytes. But in case we want to increase the amount of this fluorine group, we can also use an alternative polymeric approach. So we can synthesize this fluorinated hyperbranched polyglycerol, which contain these F3C groups. This was done by simply ring opening polymerization, multi-branching polymerization involving glycidol, as well as this custom-made trifluoroetoxymethyloxyrin. So we have a statistical distribution, you see, of this fluorinated group inside, and in here we can see a phantom image done by fluorinated MRI with the signal-to-noise ratio, which depends not only on the amount of polymers, but also on the architecture of the polymer. So how, of course, dense this grafting is and how distant each of these F3C are among each other. Then we also have to consider that once we reach then cells, we need to trigger specific interaction for an effective drug delivery. So in this case, we can still play with the polymer architecture in order to achieve specific polymer functionality. And by having a multifunctional polymer, clearly we can bind specific receptors on cells. We can also achieve clustering of this receptor in order to enhance specific cell signaling. And in this example, I can show you a polyglycerol methacrylate, which was synthesized still by atom transfer radical polymerization, which was further functionalized with these thiomannose, which can be linked to the acrylate very efficiently by microtype addition. And then we also attach a fluorophore to the terminal group of this polymer by using the standard click chemistry, as you see. And we demonstrated that by varying the content of mannose in the polymer, we can enhance the uptake of this nanomaterial. And the mannose has to be definitely linked to the polymer in order to achieve the right cell binding. For example, when the receptor was inhibited by a calcium sequestrant agent, clearly we couldn't see any internalization. So again, by dealing with the functionality of the polymer, we can trigger specific cell interaction. In this case, this was dendritic cells. In a more recent study, we attached the mannose through click chemistry into these complex four-arm architectures, which are similar to the micelles I was presenting before. And this polymer, depending on the how many mannose we have, so the valency of mannose per chain, we can achieve binding of this concanavalin A, which is another lectin binding protein. And the best binding was obtained with 2 mannose per molecular side chains. Finally, I would like also to show how the grafting of an additional peptide to this complex architecture can be useful for interacting with the specific cells. For example, in this study, we evaluate interaction with this nanomaterial with the glioblastoma cells. And this peptide is a penetrating peptide which can be activated only when the metalloproteinases are expressed by tumor cells. When the MMP is overexpressed, this part of the peptide is cleaved, and therefore the final sequence of penetrating peptides can be used for entering the cells. And in this last slide, I show you how these particles were able to deliver doxorubicin in the nucleus of glioblastoma cell lines, where standard human endothelial cells were not able to accumulate doxorubicin because this peptide was not able to be cleaved by the metalloproteinases that these cells do not produce extensively. So finally, let me conclude, say that with macromolecular engineering, we can definitely do a lot of things nowadays. So by designing the polymer architecture and the functionality, we can induce different performances. With the protein therapeutics, there's still a lot of research going on, particularly regarding the possible intracellular delivery of proteins. There's still a long journey to do in order to achieve final cell targeting for an efficient tumor inhibition, by the way. Thank you very much for your nice conference.